Hi, my name is Kevin Wong, and I'm the director of the Desert Institute at Joshua Tree National Park, which is the educational program of the Joshua Tree National Park Association. With me is Yanni Adao of the National Park Service. We will be your hosts and the producers of these webinars. Before we get started, I'd like to offer some tips on how to use the webinar effectively. As internet bandwidth is being heavily used in California due to the stay-at-home direction, and so many of us are working remotely, we may have issues with internet connections. If you have audio or monitor issues during this webinar, it may be due to having too many programs open, including your email or Facebook, which have regular updates. These will reduce your access to bandwidth. In addition, when the webinar begins, you will see a small control panel on your screen. There is a question mark and a dialog balloon. If you click on the question mark, you can ask questions that we will present to the speaker during his presentation, their presentation. We will also unmute the audience at the end of the presentation so you can ask questions. At this time, we will begin. Today's presentation is Joshua Tree Genome Project with Chris Smith and Jeremy Yoder. Chris and Jeremy, thank you for being here today.
Thank you.
Thank you.
Okay, good afternoon, everybody. Oh, I can actually see the attendees if I come down here. I appreciate everyone being able to attend, quote unquote. And today I'm going to be telling you about a project that my collaborators and I have been working on getting off the ground for the last few years. And we've had some recent significant liftoff. But I'm going to start out by just telling you a little bit about the project. I'm going to give you some background on the problems we're trying to solve, many of which I think will already be very familiar to you. And then Jeremy will tell you a bit about some of the methods and the research tools that we're using. And then at the end, I'm going to come back and tell you a little bit about where we are in the project. The Joshua Tree Genome Project is actually a large collaboration between a bunch of different scientists with many different areas of expertise. These include Todd Eskew and Leslie DeFalco, who are both scientists at the U.S. Geological Survey and who've been working on Joshua Tree ecology, amongst other things, for nearly 2 decades. Todd is best known for his work on desert tortoises, but has also done a lot of work developing community science projects across the Mojave. Leslie's work is on restoration ecology, and Leslie has done some of the work looking at the effects of the fires on Joshua Tree in the national park. Jim Liebensmack is a professor of plant biology at the University of Georgia and is also a fellow at the American Academy for the Advancement of Science. Jim is kind of the fairy godfather of plant genomes, and in particular yuccas. His students, Carlina Hayduk and Michael McCain, are now both in their starting faculty positions. Carlina is a professor at the University of Hawaii. She works on the evolution of CAM metabolism, or carbon fixation in succulent plants. That's very common in desert environments. Michael McCain studies genome evolution with a particular focus on the Agavoideae, so the subfamily that contains yuccas and agaves. And my collaborator and co-PI, Jeremy Yoder, who's with us in the webinar, is an assistant professor moving more rapidly than he might like towards tenure at Cal State Northridge, and sort of our local representative. And Jeremy's work is on using association genetics to identify genes underlying adaptive traits. Okay, and so I think all of these stories I'm about to tell you are going to be pretty familiar, but no presentation about Joshua Tree would be complete without telling these 2 absolutely obligatory stories. We know this actually as a historical fact, that the first European explorer to see Joshua trees was the Army Major John C. Fremont, who later went on to be appointed as governor of Arizona, and I learned from Wikipedia this morning had a rather dodgy career. But one of the things he's best known for is mapping the Oregon Trail and mapping the Mojave Desert. and Death Valley. And he famously describes crossing from Cajon Pass into the area that is now Palmdale, and describing how there's this sudden appearance of yucca trees. And he says, associated with the idea of barren landscapes, their stiff and ungraceful form makes them to the traveler the most repulsive tree in the vegetable kingdom. And of course, all of you know probably the possibly apocryphal story that Mormon settlers traveling along the Mormon Trail from Utah to to California outside of Los Angeles saw in the silhouette of the Joshua tree the prophet Joshua pointing the way toward the promised land. You can see out here Joshua's outstretched arm. But my interest in Joshua tree actually stems from my work on pollination biology. So Joshua trees are part of a truly remarkable pollination mutualism. As many of you probably already know, Joshua trees are pollinated exclusively by yucca moths, with typically one species of moth for each species of yucca. The moths exhibit this highly unusual and highly stereotyped pollination behavior. So what we're looking at here is a cross section through a flower. This is a petal, and this is another petal. They're forming this kind of tube here. And down inside, you can see this female yucca moth. She's perched on top of the pistil, or the female part of the flower. And right under her head here is this ball of pollen. And she's using these uniquely derived, so not present in any other insect's tentacles, to actually pull out little bits of pollen and deposit it directly onto the stigma, or exactly in the spot that it's supposed to go. And I've never been a yucca moth, but it sure looks to me like she's doing that on purpose. And one reason we think that she may be doing that on purpose is that immediately after laying that pollen, she's going to deposit some eggs into this style, so into the top of the flower here. Those eggs are going to hatch into caterpillars that will eat some of the seeds. And because yucca moths are the only thing that pollinates Joshua trees, in order to ensure that there's food for her babies, she wants to make sure that pollination happens. And this pollination system is so unusual that Darwin famously described it in a letter to J.D. Hooker, the curator at the Royal Botanical Garden Kew, as the most wonderful case of fertilization ever published. So, you know, we got Mormon settlers, we got John C. Vermont, and we got Darwin. What is not to love? I'm coming at this from a perspective of, one, to understand the pollination mutualism, but it turns out there's a lot more that we can learn. And some of the applications of this project may be to conservation biology. So as many of you already know, Joshua trees seem to be disappearing from the park. Work that was done by Cameron Barrows, Cameron Barrows at UC Riverside, suggests that over many large portions of the park, there are no juvenile trees, there are no seedlings being formed. And in the areas where there are seedlings, those are areas that are forecasted to be regions in which Joshua trees will persist under some climate change models of expected changes over the course of the next decade. And so it seems as though the effects of climate change are already playing out in Joshua Tree with this reduced region of recruitment. In addition, some recent work that was done by Juniper Harrower, who is a PhD student, or former PhD student, now alumna of UC Santa Cruz. She looked at some elevational transects within the park, and what she found was that there's this sort of narrow elevational band between 1,000 and 1,600 meters, or between 3,000 and 5,000 feet, where you actually do find seedlings. Everywhere else, all the reproduction seems to be through cloning. And that region also corresponds to the area in which there are yucca moths. And Juniper has argued that what we're seeing here is that there's an upper elevational limit on the areas in which pollinators can occur. I have some unpublished data from last year showing that actually you can have very high seed set at quite cold environments, so up in the San Bernardinos up to almost 8,000 feet. Last year we found trees with abundant fruit, but this was Juniper's finding and does suggest cause for concern. And as probably many of you saw recently in the LA Times, the California Department of Fish and Wildlife has recommended that Joshua trees be considered for listing under the California Endangered Species Act. So we're approaching trying to deal with both these ecological questions about pollination biology and these conservation issues from a genomic approach, or by understanding the Joshua tree genome. And so quickly, a genome is just an organism's complete DNA sequence, including all of the genes as well as non-coding regions. You may have heard previously about the Human Genome Project. This is comparable, but for Joshua trees. And just as a reminder, DNA is the code of life, by which we mean that DNA contains the blueprints for making genes, or it has sections of it that contain important information. And that information includes instructions for making proteins. Proteins are the building blocks of cells and everything, most things in our bodies, including, for example, hair. This is the protein keratin. Those proteins are made up of smaller molecules called amino acids, and DNA contains, inscribed in these three-letter words, instructions for stringing together amino acids to make different proteins, like keratin or lactase that we do use to digest sugar. DNA also contains non-coding regions, which potentially contain instructions for gene regulation, or telling the plant what proteins it should make, when, in what parts of the plant, and in response to which environmental conditions. So for example, perhaps there are some genes that are turned on in response to drought or heat stress. And so actually any genome project is actually a genomes project. So we're going to be generating data eventually from thousands of individuals, and we're going to be looking for genetic differences in the genome of each of these different organisms. And by looking at those genetic differences and doing some statistics, we can potentially identify regions of the genome that might be involved in important features that we care about, like adaptation to yucca moss, or differences in trunk size between Yucca brevifolia and Yucca jageriana, or maybe genes that are involved in response to drought and heat stress. And Jeremy is now going to tell us a little bit about how he's using some genetic tools to answer those questions.
Yeah, thanks, Chris. So let me, I've got the screen shared. Here we go. So genetic data has been used in conservation biology actually for a while now. Some of the things I'm going to talk about have been in practice since before I started my PhD. But in general, we can talk about sort of four ways that genetic information is useful in a conservation context. And these are kind of stepping up in order of complexity and in order of they're getting more complex and less established. So the most common, least complicated way is just assessing variation of populations. And next in this hierarchy is delimiting populations and species. So figuring out what things are the species or the population that we're interested in, defining conservation units. Then moving a little bit more complex, we might want to assess not just variation, but adaptive variation. So variation that can allow a population to cope with changing environmental conditions. And finally, the most cutting edge application that we've got so far is prediction of adaptive traits. And this is something that is not widely done, but I'm going to talk about an example of it as we go forward. But let me just walk through each of these in a Joshua tree context, and hopefully you'll see how they build up to what we're hoping to do with the Genome Project. So first off, assessing a genetic variation. And this is just answering the question, is this population of things that we care about diverse enough to avoid inbreeding depression? If a population is very genetically uniform, if all of the individuals in the population are very genetically similar, there's an elevated risk of genetic disease and genetic disorders. And that's not necessarily completely related to the number of individuals in the population. So just counting individuals doesn't necessarily answer this question. And what we can do to tackle this is collect DNA sequence from a sample of individuals in the population. And of course, Joshua trees, like humans, like lots of other eukaryotes, we all have 2 copies of every chromosome. We're diploid, and so each individual will have two sequences, one for each chromosome. So you collect some DNA sequence from the same part of the genome for every individual, and you look for places where some individuals differ from other individuals. So you can see in this case, we've got a point in this sequence where tree 1 and tree 2 have A's, and tree 3 and tree 4 have G's, and another place where tree 1 where there's some variation in which trees have T's and which trees have C's. These points we call single nucleotide polymorphisms. Each of these DNA letters is a nucleotide, and there's only one letter that defines the feature. So there's single nucleotide and polymorphism. They vary, and the different versions of these SNPs, we call them, are alleles or variants. So we would say the A allele at this first SNP, or the G allele at this first SNP. And this kind of data answers our question. This is a population with a little bit of diversity. We might want to look at more SNPs than just 2 to make that assessment, but this is basically how this kind of analysis works. Very, very closely related to that is just delimiting populations or species, and that's a question like, are individuals from this site part of the same population as individuals from that site? Or is this population even sufficiently different from that population to require separate protections? Do we need to handle these as though they're separate species? And this is, again, something that we can do just by collecting DNA sequence data from different individuals at the different sites. And you may see my Joshua Tree cartoons here reflect what we recognize as the eastern and the western Joshua Tree types. The western is the one that you have in Joshua Tree National Park. But again, in this case, we now don't just look for SNPs that show variation between individuals, but we look for SNPs that differ between the 2 possible separate populations. And so in this case, we've got some variation. At the first SNP, all of the trees at site 1 have an A, and all of the trees at site 2 have a G. But at other SNPs, we've got cases where there are alleles that are shared between the 2 sites, but they're at much higher frequency, or they're at different frequencies in one site or another. And putting together information like this from multiple SNPs, we can do things like calculate a probability that an individual tree belongs to one population or another. And this is a very common analysis in population genetics and conservation genetic contexts nowadays. This is and this slide is published data from Joshua trees. So it's a bar chart with very narrow bars, where every bar represents one of about 400 Joshua trees from about 28 different sites. The sites are separated on the x-axis by these little vertical bars. And the bar for each tree is colored according to whether we think it belongs. A clustering algorithm thinks it is most probably belonging to one population or another population. And those are a little arbitrary, but I've colored them blue and green to correspond to western and eastern populations. And what you should see here is that trees from the western sites are mostly blue, and trees from the eastern sites are mostly green. And we have a contact zone where the two types of Joshua tree come together, where there's a bunch of trees that are partially blue and partially green. And there's a lot of individuals even in the western and eastern sites that are far away from that contact zone that are ambiguously assigned too. But this is this kind of data is part of the reason that we have started to think about Joshua tree as being at least 2 separate types and possibly 2 separate species. and we might want to treat them differently. The third thing, and this is starting to get more advanced, is not just asking if a population is genetically variable, but asking if the population will survive an expected change to its environment. And this is really relevant for Joshua Tree, but for lots of other cases where we are anticipating bad effects of climate change. And in evolutionary biology, we have a bedrock principle of how evolutionary change occurs via natural selection that answers this question to some degree. If a population varies for a trait and that trait has a genetic basis, then there can be evolutionary change. So take a cartoon example where we have a population of trees that are orange and blue. If natural selection occurs that is related to those colors, so that blue trees are more likely to survive, or blue trees make more offspring, or maybe they do a combination of the two, in the next generation, we expect to see more blue trees. That's natural selection. And mathematically, we in evolutionary biology, we express this using a formula that is one of the simplest that we have. There's just two letters to keep track of in this. One of these is P, and P is the frequency of a genetic variant in the population. So an allele of a SNP, say. Delta P is change in the frequency of the variant. Just does natural selection make the allele more common or less common over time? And that's adaptation. S, the only other letter in this equation, is the strength of natural selection. And this other term, which is a function of the allele frequency, that's genetic variation. That's how much individuals in the population are similar or variable. And you don't need to do a great deal of algebra to see that basically if this blue chunk of the equation gets bigger, the other side of the equation will get bigger as well. The total term gets bigger because this is multiplicative. So basically adaptation, the result of this equation, happens faster if there's more genetic variation in the population. And by the same token, if the blue part of the equation is equal to 0, if there's no genetic variation, adaptation can't happen at all. To go back to our cartoon, basically if you have blue trees and orange trees, you can have natural selection that gives you more blue trees. But if all of the trees are orange, it doesn't matter how much of an advantage the blue trees have. There's not going to be blue trees in the next generation. So to assess adaptive variation, evolutionary theory says, well, the population will survive an expected change to its environment if there's variation at genes involved in adaptive traits. And that's an awkward answer because it tells us that we can assess adaptive variation if we know genes for adaptive traits, but how do we know what traits? And how do we find genes for those traits? And both of these take experimental work, and they take genetic sequence data, and more genetic sequence data than we need for either just assessing variation or delimiting populations and species. So what we can do though is if we have information on trait variation and genetic data from the same individuals, we can put them together to find out what genes are responsible for those traits. So take some more cartoon trees, and we have measurements of their trunk height, how tall they grow before they start to branch. And let's say we have that SNP data again. We can group individuals based on which alleles they carry for each SNP, and ask whether they differ in the trait that we've measured. So for example, each of these points represents a measurement from a single tree, and they're grouped based on what alleles they carry at SNP1. You can see here there's no difference. The range of trunk heights is pretty similar regardless of the SNP alleles. But here at SNP2, there is a relationship. If they have individuals carrying 1 copy of C have more, and individuals carrying 2 copies of C have even more, or even greater height. And so we would say there's a significant relationship there that that SNP2 is, excuse me, I'm going to try to prevent this jumping around on me. I think that'll do it. SNP2 is associated with trunk height, with the trait that we've measured. And that's great, but how do we know what SNP that we want? How do we know that we're to sequence the right place? Well, the trick that we do for that is to collect data from as much of the genome as possible. This is a cartoon of a chromosome, and ideally we would do what we call genome-wide association studies, where we look at SNP alleles at as many different places in the genome as we possibly can, and relate those to the trait that we're interested in, and look for the cases where we see significant associations. In a lot of them, we don't expect to see anything, and in a subset of those, we would. And we infer that those are places in the genome that contribute to the trait that we've measured. And this is where we start to need resources like a genome sequence, because we need to be able to place all of these points in the across possible places in the genome that could contribute to traits that we might be interested in. And then if we know what those associated SNPs are, we could go back to our populations that we're interested in and look not at any SNP, but only at the SNPs that are associated with the trait that we're interested in. So if that was a trait like seedling drought resistance, that would be something we might want to assess in Joshua trees. The catch with this yet still is that GWAS tells us what markers to use for assessment, where to look in the genome to track adaptive variation. But to do GWAS well, we really need to use measurements from controlled settings. So collecting data from trees just in the wild is challenging because there are confounding factors. Trees in different places in the desert may experience different environments. They could differ in their trunk height, not because they have different genes, but because they are experiencing different conditions. And traits that we might want to know about, like seedling survival, those might be much harder to measure in natural populations. It's easy to go collect data from standing Joshua trees, but what we're concerned about, right, is recruitment of young Joshua trees to replenish the populations. And so what we'd actually like to do is be able to do GWAS in controlled settings in seedlings. And fortunately, seedlings are amenable to this in greenhouses or experimental gardens, and use the results of GWAS to predict those traits in natural populations. And this is kind of the next frontier for conservation genetics. It's really not, there are not a lot of species that have the genome sequencing that's been done that's necessary to support this. But I actually have worked on one of the projects where this has been possible. And this is something called adaptation. So this is a project out of Canada, based at the University of British Columbia, that collected a whole bunch of genetic data for lodgepole pine, about 30,000 single nucleotide polymorphisms from almost 3,500 trees at 280 sites across British Columbia and Alberta. And a subset of these were seedlings that were grown up in growth chambers and measured for a whole bunch of climate adaptive traits, like bud break and bud set. So when do they start growing in the summer, and when do they harden up for winter so they don't freeze to death? What the timing of their total growth is, how prone they are to cold injury. You can take a plant and zap it with freezing temperatures and measure its physiological the physiological damage that it takes and how badly it's hurt. And so this is still actually unpublished data, but this is one thing we can do with this combination of information. I performed GWAS using these growth chamber measurements to find SNPs that were associated with all of these different measurements that we could take on the seedlings. And then I took that back to all of the other trees that we had genetic data from. And we didn't have traits measured in these trees, but because we had genetic data, we could predict what their traits would be if we had taken those measurements. And we call those genetic values or predicted traits. And this is what I think is a pretty cool result. I can look at how those predicted traits relate to the environments that those other trees were in. And we see patterns that are consistent with what we would expect. Specifically, so this is one of the nicer examples. This is the genetic value or the predicted trait value for that cold injury risk. So how badly a seedling is hurt when you freeze it. And trees in populations that were that were warmer and wetter, up at the high end of this scale, had higher cold injury risk. They were more prone to be damaged when they froze than trees from colder, drier conditions. These trees are predicted to be better able to support freezing, and that makes sense. We would expect that genetic differences in this trait would exist between populations that are experiencing different climates. And this scale over here is an estimate of how statistically significant that relationship is, and it's much stronger than we would expect by chance. So the goal of the Joshua Tree Genome Project is actually to get to this point with Joshua trees. We're able at this point, we can do assessment of variation, we can delimit populations and species with Joshua trees pretty easily. We're just getting to the point of building up the resources to assess adaptive variation and predict adaptive traits. And the project that we're really just getting off the ground that Chris was leading in on is what's going to get us to those points. The overall goals are we're going to complete what we call a reference genome for Joshua Tree. So that's a genome sequence from a single individual that we sequence in great detail, and we identify which parts of the genome are coding for proteins and which parts aren't. And we're going to test Joshua Tree seedlings in experimental common gardens all over the Mojave Desert. And we're going to use those seedlings to identify loci that are associated with climate adaptive traits in the seedlings, and use the results of that to predict climate adaptive traits of natural Joshua Tree populations. So by the end of all this, we should be able to go to a Joshua Tree population and collect some genetic data and tell you whether or not the population has the genetic potential to cope with the climates that we expect that population to have to deal with in the next 50 or even 100 years. And with that, I think I'm ready to hand off to Chris.
So, I think I've kind of a tough act to follow there. After Jeremy effectively described all of conservation genetics and evolutionary biology and population genetics in 20 minutes. But I'm going to try to tell you a little bit about where we are in this massive project that you just heard about. And so to give you a sense of the scale of the problem, I wanted to describe the size of the Joshua Tree genome. So the Joshua Tree genome is estimated to be about 3 billion bases in length, which that's about the same size as the human genome, which famously took 10 years and $3 billion to sequence. But maybe to give us a more familiar and tangible reference, that's about 500 times the length of the complete works of William Shakespeare. I went through and used my computer to actually count up all of the characters, including spaces, in the complete works of Shakespeare. And it turns out to be about 6 million characters long. The Joshua Tree genome is 500 times that. And so that seems like a just impossible task. But due to some really amazing advances in, I'm seeing 335 there. Okay. Due to some really amazing advances in technology that have happened in the last decade, particularly the advent of something called Illumina sequencing or next generation sequencing, our capacity to generate sequence data has improved dramatically, and the costs have reduced at a staggering rate, such that it's now possible for us to generate about 120 billion bases of DNA sequence data for about $3,000, and that takes about a week. Some of you may already have followed this project in the past, but using the crowdfunding site experiment.com, we were able to raise more than $10,500 towards sequencing and getting a pilot project going. And in addition, the Living Desert Zoo and Gardens gave us a very generous gift to try and get us off the ground. And so how far have we come with that money? Well, so far we've sequenced about 105 billion nucleotides of data, or we've sequenced the Joshua Tree genome about 35 times over. And so it might seem like that is the time to open up the champagne and say eureka, but unfortunately, that data comes to us in fragments that are about 150 characters long, or it is though we had taken all 500 of those copies of the complete works of Shakespeare and run them through a shredder. And this is not something that we did wrong, fortunately. This is actually just a fundamental feature of the Illumina sequence data, is that it comes in these small little fragments, these small little bits. And so our task now is to take these little 150 nucleotide pieces and try to figure out where they go. We have a bit of an advantage that since we've sequenced this 35 times over, and since the fragments are random, we can potentially line them up. So we can look at this, his fell to Hamlet, now Sir Young Fortinbras, and see, oh look, this matches up here, Sir Young Fortinbras. With 500 copies of Shakespeare, we're not doing that ourselves, we're doing that by the computer. And computers are pretty smart these days, so that also might seem like, okay, simple, straightforward task. Unfortunately, again, it turns out that the Joshua Tree genome and many genomes, including the human genome, are filled up with these regions called tandem repeats, where the same little bit of information is written over and over and over and over again. You might think about this as being comparable to the scene in The Shining, where Jack Nicholson's character loses his mind and types "all work and no play makes Jack a dull boy" over and over again. And so to deal with that problem, we've been turning to a newer, like cutting edge, bleeding edge technology called nanopore sequencing that uses changes in electrical current as a fragment of DNA is pulled through a hole to try to sequence the data. And doing that, we use these tiny little DNA sequencers that actually fit in the palm of your hand. They're smaller than an iPhone. They cost about $900, and out of those we can typically get about 30 billion bases of data, and those come in size pieces that are not 150 nucleotides in length. They come in pieces that are sometimes up to 1 million bases long. So that seems to be way, way better. However, and so far we've generated about 14 billion bases of nanopore data. So together we've got, everybody good? I'm hearing some surface noise. Okay, together we've got about 130 billion bases of data. The problem with the nanopore data is that it has a very high error rate, about 30%, and so that is a downside of the nanopore data itself. But what we're working on right now is doing what's called a hybrid assembly, in which we take that potentially error-ridden nanopore data, which here might say to lie, to sheep, to leap, perclance to cream, and we can correct that with the Illumina data. So we come up with something that says to lie, to sleep, perchance to dream. So at this point, we're taking about 120 billion bases of data from these different sources and putting them together. And my colleague Mike McCain is working on generating even more nanopore data. And we're also turning to something called PacBio data. Okay, so that's where we're at with the genome. Jeremy was also telling you a bit about trying to infer adaptive traits. And to do that, we're working with this Mojave Desert Common Gardens project. This is a project that it was put together by the U.S. Geological Survey, and particularly Leslie DeFalco. And what Leslie has done is to identify sites at different regions throughout the Mojave that experience different climates. And the goal of this project originally was actually to do some local adaptation studies on native desert plants for restoration work. So, if you have an area down near 29 Palms that for some reason has been hit by 100-pound bombs and you want to restore the plants there, you know that you want to use the seeds that are well adapted to this area. And her common gardens span the range of Mojave Desert climates from 29 Palms and Ridgecrest up to St. George or Pahrump. And so we're taking advantage of these common gardens as a natural laboratory, right? Laboratory, this is not that natural. And what we've done now is to collect Joshua Tree seeds from sites across the Mojave, encompassing the range of climate variation. We have sites now, tree seeds now from 16 sites and 10 matrolines, or 10 different trees at each site. And this summer, we're planting 1,600 seedlings into the greenhouse, which then will be transplanted in the common gardens. So then we can then do those growth, survival, and physiology measurements over the course of a 4-year project. And by putting those together with the genetic data, as Jeremy was describing, we can potentially identify genetic variants that are involved in adaptation to climate. And lastly, we've undertaken a large project to compile data on the demographic distribution of Joshua trees in different sites across the Mojave. Using some randomization software, we've identified sites that are on Bureau of Land Management land and are very likely to contain Joshua trees based on some modeling work. My student Patrick Baird has then gone in and zoomed in on Google Earth to every single one of these sites and made sure that there are actually Joshua trees there. And we're now collecting data from those sites. And the ultimate goal is that we'll be able to pull out of that information about which of these sites contain the genetic variants that are likely to allow them to adapt to climate change, and which of these sites show demographic profiles where it looks like they're actually producing seedlings. So those might be like those high elevation sites up in the San Bernardinos, or those high elevation sites up in Joshua Tree National Park. And we can say, okay, if we want to spend X amount of conservation dollars, we want to invest in preserving, say, this site right here that has the necessary genetic variation, its population is healthy, and it looks like it's going to be able to adapt. But this site over here, it doesn't have the genetic variation it needs, and it's already on its way to extinction, so let's not deal with it. And as a way to try and collect data from all these sites, we're working with a network of community partners, including the California Native Plant Society, Transition Habitat Conservancy, which is outside of, in Pinion Hills, California, Mojave Desert Land Trust, and we're recruiting groups of volunteers who are trained to collect demographic data for us, and they then lead groups of their associates to go and collect data. Getting this part of the project has been a huge lift. The idea was supposed to be that there could be data collection going on that didn't require me to fly from Nevada to Palm Springs every single time, but it's actually been fairly slow to start off. And what we really need at this point to make this part work is program leaders. So people who are excited about taking a leadership role in recruiting volunteers and leading groups to go out and collect data and doing social media to get involved. And so if you think that's something that you're interested in, please get in touch with me, because the program needs you. Lastly, I just want to let you know, and we're a little bit about 5 minutes over time here, that we are expecting, that I want to let you know that we have a website where you can follow project updates. And I want you to watch this space, because we're getting ready to make a very big and exciting announcement sometime, hopefully in the next month. And with that, sorry for a somewhat rushed presentation and some technical problems, but I want to turn it over to any questions there may be.
Okay, at this point in time, let's unmute all the participants on my end, and then you will be able to ask questions. Just check and make sure that you are personally unmuted, and let's open this up for questions from the audience.
This is Dave Larson. So I'm wondering, how much material do you need to collect Joshua Tree in order to figure out what his DNA is?
You can hear me now. Jeremy, can you wave at me if you hear me? Okay, so a lot of the work that we're doing is done in my lab. And the material that we take out to get the genome is actually a very small amount of DNA. It's often a microgram or 10 micrograms, so 1/100th of a gram of DNA, and that easily could come from a single leaf. In practice, we're collecting many times that. So the nanopore sequencing that I was talking about requires very high quality DNA, and to get that, you sort of start with often when we're doing that in the lab, we're using a whole seedling, and Mike McCain's lab has perfected some techniques to use a razor blade and peel off the very outer green layer, avoiding all the fiber. For the purposes of this resequencing that Jeremy was talking about, where you go and get DNA from the wild, it's often done from 5 leaves or less, and usually we're using 1/10 of one leaf, and then we have those other 5 kind of as backup. And so
A couple of typed in questions. One is, how often do mutations occur that allow for species to adapt?
Okay. Actually, I have a paper that is in revision that is specifically about estimating mutation rates. And I'm going to give a not very helpful answer. But what I've found is that the rate of mutation in the Joshua Tree genome is pretty comparable to the rate of mutation in humans, which in general means not very fast. But because there is so much DNA, every individual, every time they have a baby or every time a Joshua tree makes a seed, every individual probably carries some new mutations. So for example, it's estimated that every human that's born contains about 10 genetic differences that were not present in either of the parents, 10 new mutations. And so what you do need to get lucky in that you need to have genetic variants that are present, so mutations that are present in the region of the genome that's important. And if Joshua trees are going to make it, so if they are going to win this kind of genetic lottery, probably they're going to need to be relying on mutations that are already out there existing in the population. So this is where Jeremy was talking earlier about how you have to have genetic variation. If we have to wait for new genetic variation to be formed, I was about to drop an F-bomb, but they are in bad shape.
Another question, do Jeremy and Chris see protein amino acid analysis playing any role in the genome project?
Jeremy, do you want to speak to that? Are we looking for coding changes, or are we looking for non-coding changes?
Yeah. So one of the components of building a reference genome is, like I said, figuring out what parts of the genome are specifically coding for proteins. And the way that's done is actually to read the DNA sequence that you've come up with and look for. So there are specific codes for every amino acid. There are also specific codes to start and stop reading DNA at the beginning and the end of a protein. And so among other things, you look for start and stop codes in the DNA and say, oh, this must be a gene. And yeah, one of the things we can do is reconstruct from the DNA sequence what the amino acid sequence and then what the protein would be. And part of understanding what the function of a gene is comes from that kind of analysis. I think Chris has something he wants to add.
I might be misunderstanding the question, but I think the question was about, are amino acid changes important, or are regulatory changes important?
Well, so yeah, if the question is what are we looking for, could be both. One of the so regions outside the protein coding sequences are where we find regulatory sequences, so regulatory elements that control when a protein is made versus not. And so you can imagine that if, say, a protein is unhelpful, if it would be beneficial to stop making the protein, a mutation that changed the regulation so that it was just never produced would be useful, or a mutation that broke the protein so that when transcription occurred, it stopped. That could have the same beneficial effect. One of the ways we make it practical to collect a lot of DNA sequence data for Joshua Tree, we think, is going to be to focus specifically on the protein coding parts of the genome. That's a much smaller portion of sequence than the whole genome itself, and it makes it possible to get data from thousands of individuals for the kind of budget we can get from a single round of grant funding. Yes, the answer I guess is that we're mostly going to be looking at protein coding sequence in a lot of this going forward.
So one of the things that Jeremy wisely omitted, even though I was like, oh, we have to talk about that too, is part of the Common Gardens project is looking at what's called gene expression data. So Carlina Hayda, her specialty is in looking at gene expression. And at these different points in time when we're measuring drought stress and things like that, we're able to look at what genes are turned on. And yep, that's my answer. I'm sticking to it.
Okay, the final one is not really a question, it's more of a statement. It says, thanks for the presentation. We should touch base again with Chris and Jeremy about collaborating with the park. Desert Institute side, I see an opportunity for us to do a collaboration. We can perhaps provide an audience for you for volunteers. So maybe if you want to train some program leaders within our team, we will reach out and find some volunteers and actually conduct regular programming, much like we've done with Chris in the past, and help you in that aspect. So let's talk about that in the future.
Okay.
We'd be really excited to do that.
I'd love to talk to you, Kevin, and actually if we could follow up maybe shortly, just right after this meeting. I have a couple thoughts and questions. Thanks everybody for your attention. I know that was jam-packed with information, so I hope it was helpful to you. And good luck to everybody during this extraordinary time. Please do your best to keep healthy so that we can see you all in the park.
Chris and Jeremy, thank you for being here today. And thank you to our audience. All of our webinars are recorded and can be viewed on the Desert Institute YouTube channel. In addition, if you're interested in learning more about the platform we use for these webinars, please let me know. Our next webinar will be on Wednesday, April the 29th, and we have Kurt Leuschner, Amazing Desert Insects. Until then, please stay safe and healthy. Thank you.